Good morning, everyone. I'll be giving a presentation on the topic carcinogenesis. I'm Dr. Sandeep Sapkota. I am an NBMS subspecialty resident at BPKMCH. Starting with the introduction, I would like to start with the introduction of the neoplasia. The most accepted definition is given by Dr. Willis in 1952, where he defined neoplasia as the abnormal mass of tissue, the growth of which exceeds and is uncoordinated with that of the normal tissue, and the growth persists in the same excessive manner even after the cessation of the stimuli which evoked the change. And the next definition I would like to talk about is of carcinogenesis. It is defined as the multi-step process where the normal cells undergo genetic and epigenetic changes which ultimately leads to the uncontrolled growth and malignant transformation of the cell. And now diving deeper into the topic, the three major components of a normal cell to form into the fully grown malignant metastatic tumor is carcinogenesis, oncogenesis, and then metastasis, where the carcinogenesis means the initiation of tumor, oncogenesis means the continuation and maintenance of the tumor, and metastasis means the spread of the tumor. There is always some overlapping component between these three, but the talk about metastasis, I'll keep it out of the frame because I'll focus mostly on the carcinogenesis and oncogenesis. Starting with the historical timeline, if we see the word cancerous or word related to the tumor was mentioned in 1700 BC in the Babylonian Code of Hammurabi, Chinese rites of Zhou dynasty, and even in our Indian epic Ramayana. And then in late 17th or 18th century, Percival Pott, an English surgeon, he linked a scrotal cancer to chimney sweepers caused by chimney sweep. In 1858, Rudolf Virchow gave the cellular origin of the cancer. In 1917, Amigawa et al. From the Japan first did the experimental proof leading from a carcinogen to the cancer, the paper of which I'll present in the next slide. In 1937, NCI was established, which helped in boosting the research and robust framework for the treatment of the cancer. And in 1973, Nordson gave two-hit hypothesis. And in 1983, Bremblom and co. they defined the concept of stages of carcinogenesis. The modern theory of the carcinogenesis was given later by Hart and Turturro, which I'll discuss later on. But this paper from the Bremblom and co. was the green signal for the treatment of the not just treatment, but understanding of the cancer as a whole. This is the series of the study proposed by the Amigawa et al. From the Japan. They painted the coal tar in the rabbit ear, and then after 8 years, they saw that they developed the squamous cell carcinoma, which was the first experimental proof of the cancer. So after some of these historical snippets, I'd like to talk about the carcinogenesis theory that have been put forward. The first one is genetic mutation somatic theory, which is and then epigenetic theory, and inflammation and microenvironment theory on inflammation and microenvironment, stem cell based theory, and multi-stage theory. So starting with the genetic theory, I'll discuss on detail about this. This was given by Dr. Hansen in 1887. We noted the similarities between gene mutation and the cancer. He proposed that there was structural anomalies in growth regulating genes, which are hereditary and transferred to progenies. And then the inflammation theory that is explains how carcinogens causes cancer. Most of these were explained in our previous topic of risk factor class. And then stem cell based theory, that is cancer cell arise from the any single progenitor cell. And then multi-stage theory. Multi-stage theory basically is what combines all of this together. And the epigenetic theory, I'll talk in detail in the coming slides. So Dr. Hansen, he proposed some evidences for the genetic theory, that is the genetic constitution, it influences the cancer risk. Some of the cancer are prevalent were prevalent in Down syndrome, and hereditary cancers, which also involve two stages, were proposed by him. These were some evidences proposed by Dr. Hansen in his published literature, and he also proposed a strong correlation between mutagenicity and carcinogenicity in his first work. So there are some causes of the genetic causes of the errors in the DNA replication, or causes of the genetic realignment, that are the errors in the DNA replication by DNA polymerase. It's an enzyme which helps in forming the primer of the DNA, ultimately leading to the replication. And then chemical DNA instability, that is depuration, or the base pairs of DNA are not aligned properly. And then free oxygen radicals, they may damage the DNA. And then failure to correct the damaged DNA, it's also the cause. These four causes were first proposed in 1857, and it has been studied in detail till now. So he also said that the single and multiple sequential mutations are needed for carcinogenesis, which will lead to the formation of the fully malignant tumor. In very short, if we see the multi-stage theory, it says that the carcinogenesis is a multi-stage process. It was proposed by Hart and Tucker in 1988, and they have given it in three phases, that is initiation of the tumor, promotion, and progression of the tumor. So it involves the stages between the DNA damage, or the first hit in the DNA, and to the full cancer manifestation. So, starting with the initiation, the carcinogens, or any genetic abnormalities, damages the chromosomal DNA, and the DNA repair usually occurs by the repair mechanism in the body, which are direct repair or mismatch repair genes, which helps in DNA repair. And if there is a failure to repair these DNA, it leads to the irreversible genetic changes. And then the second hit, that is further carcinogens like virus, radiation, they act irreversibly, and which initiate the tumor formation. And some may stay latent even without the phenotypic expression, and some may progress toward the promotion stage. If there is a chronic initiation or chronic genetic alteration, the epigenetic changes or promoters will transform the cell into the reversible phase, and if it cannot still be repaired, into the irreversible phase, which is the towards the proliferation stage, or last stage, that is progression stage. So these cells, deformed cell, undergo Darwinian computation, that is also in short known as survival of the fittest. That is, the most fit cells are the cells which can acquire high nutrients or high energy, will stay, and they will express high karyotype instability and genome mutation, which ultimately lead to the formation of the abnormal proto-oncogenes, or oncoprotein, which helps in formation of the cancer. This was the multi-stage theory proposed by. Now, I would like to start with the discussion something about the cell cycle. A cell goes through various phases, that is M phase, where this is also known as metaphase. G1 phase, sometimes it may go into the quotient phase or G0 phase, and then S phase, and then G2 phase, and then again M phase. So in M phase, a cell undergo division into the daughter cells, and these daughter cells will further go into the G1, S, and G2 phase. This is how the cell will proliferate. If the DNA is abnormal, or if daughter cells forms with the abnormal DNA, they are generally arrested at the G1 phase. G1 phase generally checks for, there is always a checkpoint inhibitor, as we can see in the picture, and G1 phase are checked for nutrients, protein, and DNA damage. And if the damaged DNA enters G1, it will take directly into the quiescent phase, and the DNA repair function mechanism will start. There are various mechanisms to repair DNA in our body, direct repair or mismatch repair. Uh, there are other system like homologous repair. I will not discuss on that. So, if a cell cannot repair the DNA, cell our checkpoint component will arrest the cell. I'll discuss it in more. And after the G0, G1 phase, the S phase, cells will be from S to G2 phase, cells will be checked for cell size and DNA replication. And from G2 to M phase, the cells will be checked for chromosomal and spindle attachment. And if we see in the small diagram here, we can see there are cell components like cyclin D, cyclin E, cyclin A, and cyclin B. Just for example, if a cell is in G2 phase and the cyclin B starts to rise rapidly, or cyclin B is free, and if it reaches to the maximum point, the cell will enter into the M phase, or cell will go into the M phase. If all of these cyclin B are consumed by the P21 and the cyclin B cannot rise to the highest level, the cell will go into the arrest. So generally there are tumor suppressor gene, that is retinoblastoma gene and P53 gene. So these are controlled basically by the E2F gene. E2F gene, they controls all the cyclin dependent kinase and cyclins and the tumor suppressor genes, and the RB genes are P53 genes. When the secondary messenger tells them to stop the or arrest the cell, they will arrest the cell. So these are some cell cycle components. The most important ones are highlighted. The P53 causes cell cycle arrest via P21. The P21 will go and bind to the cyclins, and the cyclins level will not rise, causing to the cell cycle arrest. And another is the extrinsic apoptosis mechanism via the Bach's gene, which is regulated. And also another most important tumor suppressor component is RB gene, which binds E2F to prevent G1 to S transition, as I said. So these are the gatekeeper genes, the RB gene and P53 genes. Proliferation. And to the sequence of carcinogenesis, as I said, it is a sequence from a journey from a normal cell to a malignant cell. A very good picture, a theory proposed by Ferron and Vogelstein, also known as Ferron-Vogelstein sequence. It very much discussed in the colon cancer. I am using this example as a sequence of carcinogenesis. That is, a normal cell will undergo any genetic rearrangement. That is, in this case, there is an APC gene at 5q21, and it is the first hit. It will put the mucosa at risk. And then the further methylation abnormalities. These methylation abnormalities, also known as epigenetic abnormalities, which I'll discuss. I'll come to the next slide. So the methylation abnormalities by APC or beta catenin will start the proliferation of the mucosa at risk. And then these proliferated dysplastic cells will convert into the adenomas with the effect of further proteoglobulin mutation like KRAS, with the help of the gene like KRAS. And these adenoma, they can convert into the full-blown carcinomas with the genes like TP53 and chromosome 17. The mechanism is similar to that to what I explained. And now, diving more into the molecular basis, I'll discuss more about the genetic and epigenetic changes in the carcinogenesis. So these genetic damage, it lies at the heart of carcinogenesis. This is the start. And tumor formed by clonal expansion of single precursors. The single precursor cell will proliferate rapidly and will form a tumor. There are 4 things that are required for a normal regulator gene, that are growth promoting proto-oncogenes, growth inhibiting tumor suppressor genes, genes regulating the programmed cell death, and genes involved in DNA repair. So this is the core of the tumor formation. So if I'll I'll talk more on coming slide. This is the same thing, the initiating mutation to the progression of the. So I have kept this slide for the explanation of what epigenetics is. So let's see from bottom to top. That is starting from the bottom, the right hand side of the, my right hand side, bottommost right hand side, we can see there is the starting of the DNA along with the base pairs. The so DNA is the more and more groups of DNA is now bound together by the histone protein as we go up. So the histone proteins are then collectively forming a chromatin, and then chromatins are then collectively forming a chromosome altogether. So if we see this in a cell, there are 2 meters of DNA which are bound together by the histone protein. So there are two things that we need to keep in mind to understand the epigenetics. Epigenetics means epi and genetics, that is above genetics. Now we have come above the knowledge of genetics. So in epigenetics, the first one is methylation and second one is acetylation. So if you see in the picture, there is a methyl group which binds to the cytosine group 1, and then also the acetylation group, which is also known as a epigenetic factor, which happens in the histone protein. So if a methyl or if a cysteine is replaced by a methyl group, it is known as methylation, which means the gene is silenced. So if in a p53 gene methylation happens, a tumor suppressor gene is silenced. Such as in a histone protein, if we say that it is acetylation, means the gene is activated, which means if a proto-oncogene undergo histone protein of the proto-oncogenes undergo acetylation, so the gene is activated, causing the cancer. So the knowledge of this is important because this is the basic. This is the first step of the carcinogenesis, and this is the first step where we can start our treatment if we know it beforehand. Of epigenetic. So, as I said, the epigenetic modification, it includes the DNA methylation, typically which silences the tumor suppressor genes, or histone modification, also known as acetylation of histone, which can either enhance or replace gene expression. Basically, it's the same thing, but just a deeper explanation of the tumor suppressor gene and proto-oncogenes. So these explanation has now opened the new therapeutic opportunities. The study of these epigenetic changes are clinically important for the treatment of the cancer. The epigenetic changes are termed as reversible, unlike DNA mutation, because as we said, the methylation and demethylation process can be stopped, but the aberrant genetic information that the DNA carries cannot be stopped. So this is a new information about the epigenetic therapeutic. So there are drugs targeting DNA or histone modifying enzymes, which are promising in correcting these abnormalities if we know beforehand. This is just the example of recent drugs, that is DNA methylation inhibitor, which are used in blood tumors like AML and myelodysplastic syndrome, azacitidine and desitabine. And then also the histone deacetylase inhibitor, which has been used in cutaneous T-cell lymphoma and peripheral T-cell lymphoma, vorinostat, and has shown the promising result in the studies. So, as I said, the three major things, the oncogene, tumor suppressor gene, and DNA repair gene, these are the heart of the carcinogenesis. I'll start with the oncogenes, which promotes the autonomous growth of the cancer cell, and the mutation of which will convert into the proto-oncogenes, and which will ultimately produce the oncoprotein, which will go and encode the DNA to proliferate abnormally. So this is example where a growth factor will bind to a growth factor receptor, activating a secondary messenger system, the GDP, and it will further activate the RAS system to activate the MAP kinase pathway or PITP pathway or AKR mTOR pathway. These are all the growth factor pathway, which if activated irreversibly, which if activated abnormally, will lead to the cell proliferation or abnormal cell proliferation. or unnecessary cell proliferation. So this is the same thing. And ultimately, the growth factor, these changes in the gene expression also support the cell survival and metabolism. The proteins will be encoded as the cell requires according to the new environment. So now the tumor suppressor gene. So as I said, the RB and P53, this is the core to gatekeeper genes. They help in inhibiting cell proliferation, promoting DNA repair, trigger apoptosis. I explained that RB controls the G1S checkpoint by binding the E2F transcriptions. So basically, if the tumor suppressor genes are silenced, if they are hyperphosphorylated, if they are silenced, they will not be able to check the cell cycle, and the cell will keep on proliferating. This is the uh, core concept. And the some genes that are the tumor, these APC genes, these are the most genes that we see in our clinical daily basis. That is APC gene is also a tumor suppressor gene associated with the colorectal cancer. And then NF1, NF2 associated with the neuroblastoma. PTN gene is also a tumor suppressor gene. RB gene, as I said, and CDK and QA gene associated with the pancreatic cancer. The most important one is, sorry, the most important one is the TP53 gene, that is which is encoded by the P53 protein, helps in cell cycle arrest, almost associated with all types of solid tumors. Also associated with the syndrome known as Leaf-Remini syndrome, which portrays many kind of solid tumors. So these are some of the tumor suppressor genes. CDS1 is also very important. The highlighted portion, the highlighted genes, the CDS1 associated with the gastric carcinoma, and BRCA1, BRCA2 associated with the breast cancer. These are the tumor suppressor genes, important tumor suppressor genes. So after the genetic rearrangement and genetic explanation of the carcinogenesis, sometimes the first gene can also initiate by carcinogens. This is also very important the topic while discussing on the carcinogenesis. The much of it has already been discussed in the risk factor slide. However, I'd like to put on I'd like to highlight some of these. There are three types of carcinogen: physical, chemical, and biological. Physical is a radiation, any form of radiation. Chemical means the various chemicals like nitrosamines, coal tar, the one which I discussed earlier, and the biological carcinogens like viral infections can also link to the or give the first hit to the formation initiation of the cancer. So if we see, this is the scheme of carcinogen to cancer. This is from the physical carcinogens. The physical carcinogen, they can cause the metabolic activation, which will secrete electrophilic intermediates, which will go and bind to the DNA, and which go and bind to the DNA, resulting to the DNA tension and DNA breakage, which will initiate the cell towards the cell proliferation, and which will further go and from the altered cell growth with the pre-neoplastic clones, and they'll further progress towards the formation of the malignant neoplasm. The sequence is basically same for every type of carcinogen. The physical carcinogen, they binds to the DNA directly after some electrophilic intermediate. And sorry, that was for chemical. Now physical carcinogens, uh, the various various radiations that are emitted, such as alpha particle, beta particles, gamma rays, X rays, they all has some effect on the formation of the cancer. The most telling story is that of the survivor of the uh, atomic bomb dropped in Hiroshima and Nagasaki. These radiation has a direct DNA damaging properties. If the DNA damage that is not checked by the genes that we talked earlier, will go and further proliferate into the full-blown cancer. This is the carcinogenesis sequence for the viral and bacterial and parasitic oncogenesis. Some of it will affect the p53 inhibition. Some of them will inhibit the RB, which will ultimately transform the cell into the immortal, replicating cells. And as we talk about the carcinogenesis and the tumor cells has already formed, the tumor cell attain some hallmark to remain in the body, uh, to compete with the normal cells and to proliferate themselves. So first one, I would like to start with the sustaining proliferative signal. It's an easy to it is easy if we remember the sequence. The proliferative signals can be from their own paracrine or autocrine stimulation or from growth factor. And then there is avoiding immune destruction by various genetic and epigenetic changes. They may avoid from the immune cells, the T cells in our body. They may fail to recognize the cancer cells, and they will further evade the growth suppressors. They will and then enable themselves to replicate immortality. That is, if the cell is not going into the apoptosis by these P53 and RB, they will gain the replicative immortality. And tumor will induce inflammation to stay in the body where they will get the growth factors and nutrients. Tumor will activate invasion and cell-to-cell communication. Tumor will undergo genomic instability. There will be mutation in karyotypes inside the tumor, which will further transform into different type of clones. And tumor will induce angiogenesis for more nutrients. They'll resist cell death. They'll deregulate the cellular energetics, also known as Warburg phenomena. That is, if the normal cells require glucose in an aerobic metabolism, tumor cells can live in an anaerobic environment as well. So after the hallmark, these are some of the newer topics that are being discussed in the carcinogenesis. That is cancer stem cells and clonal evolution. There is a normal cell, then mutated cell, and then clone, pre-neoplastic clone, and then subclone, and then diversity of the tumors. This theory explains the different mutation to the different site than the primary tumor. Tumor means the primary tumor and metastasized tumor are different because of the different clones in the cancer. I'd like to explain this with a simple diagram. There is a normal tissue, and their mutation has been started. So there is a P1 is a primary tumor, and M1 is a metastatic tumor. And at first, there is a shared mutation between these two. And as you see, as the clone is formed, there is a unique type of mutation that we can see in the metastasis of various sites. That you see in R6, R7, R11, there is a unique type of mutation in the various site, which means a metastasized tumor can be different than the primary tumor because of the clone and subclone theory. That theory also explains why the metastasized tumors has a different hormone status even in the breast cancer and some other solid tumors. And the understanding of these carcinogenesis has a important clinical implications like in early diagnosis. Uh, the recent concept of the liquid ctDNA, circulating tumor DNA, and the methylation, DNA methylation markers, they can help in early diagnosis, and some of it can also help in prognostic marker, that is specific mutation. T53 KRAS, they are associated with the poorer outcome and aggressive behavior of the tumor. And also helps in screening and risk assessment, the most important genetic testing, that is BRCA1 and BRCA2. And they are also they can also be used clinically in the future direction, that is targeted therapy and
we are now using drug-specific cancer mutation. Suppose in case of the lung cancer, the EGFR inhibitors are started, the immunotherapy are started as per the mutation. And there's also a concept of the personalized cancer care, the treatment tailored according to the individual tumor profile, according to the recent advances in next generation sequencing and the various mutations that we can see. We can tailor the patient as per the mutation, the treatment as per the mutation. And then the information of the epigenetics has given the direction toward the gene editing, that is correcting the cancer mutation via CRISPR-Cas9 sequence. The CRISPR-Cas9 recombination sequence has been used in correction of the genetic deformities that in the field of abnormal gene transformation in genetics, but it can be used in the cancer treatment in later time. So to summarize, the carcinogenesis is a complex multi-step process that involves genetic, epigenetic, environmental, and immune factors. The different models help explain how the cancer develops at a cellular and molecular level. The insights into the molecular and cellular level are essential for improved prevention, accurate diagnosis, and targeted treatment strategies. And this one picture, it summarizes the presentation of today, that is normal cell after the initiation of by some carcinogen or co-carcinogen will change into the normal cell at risk, and then after some promoter or direct cancerization, it will change into the cancerous proliferation, which is the core concept of carcinogenesis.
Okay, well done, Madam Sandeep. That was very well summarized from the molecular cellular processes to molecular and also the clinical implementation. That was very difficult topic. I think you did very well. So, any comments, any questions?
Thank you, Dr. Sapkota. Sapkota, like it's okay, sorry. Nicely summarized indeed, but majority of us are very, to be honest, very poor in molecular biology and genetics. This is why we don't understand these things. We don't try to understand these things also. But for us, unfortunately, we need to understand at least some basics. Let's say he tried to explain some proto-oncogenes, tumor suppressor genes, etc. And at least a few examples of those, because we are working for these patients in one hand. On the other hand, there is such an huge flood of articles that has been written, and then in every right and left you will be seeing those genes, EGFR, cytokines, or cyclins, and etc. So at least upstream regulation, downstream regulation, target area, carcinogenesis, etc. Epigenetics is a new thing that we learned today. I mean, this is not only by genetic deformation of a DNA or RNA, but some other issues, microenvironment, where the cell lies, methylation, etc., that can cause different things which can be arrested, and the cell can be brought back into the regular pathway. I mean, these are the new things that we learned today. Except indeed, the modern era of oncological development probably also falls in this category or in this area. Probably in 20, 30 years down the line, there will be plenty of immunotherapy and etc. or tiny little things that you can pass into a GI tract or in a lung, let's say, that goes somewhere and then that takes up the tumor. This probably is the future of the medicine. So we need to read these things. We need to sensitize ourselves. By his very nice summary, we hope myself and others also should go back and read again, try to understand these things. Thank you. Thank you.
Something was very catchy that he said, we assume the future will be the gene editing that he has summarized the last time. So I think that will be the future. We don't need to operate on those patients. We don't need to give the chemotherapy, etc. If something will be done right, it's not very far, I think. Everything is taking so much in this computerized data. So I think that will be very much possible because people have divided some microchips, nanomolecules that will go inside, penetrate, that will even detect the cancer cells. And that will kill only the cancer cells. So that will be the future, I think. What happens, just clinical scenario on this, okay? Why we see do the PET scan, we do the CT scan, everything we do is comes out to the stage 1 disease. But in the most of the solid tumor, even the stage 1, the long-term survival is 50% rate happening. What is happening with the rest of the 50%? Why they are going to recur, even if it's the stage 1? So nowadays, what they do this the circulating tumor DNA analysis. If the efficacy has not been well validated, but this is a very good thing. If you check that the patient has a circulating tumor DNA circulating inside the body of the patient, then what's the benefit from doing the surgery? Right? That's the question. So if something can be done at that level, and if we can control at that level, what surgery will not be required. Right? There are many things like Angelina Jolie, she is not suffering from breast cancer. She is not suffering from the ovarian cancer, but she underwent the prophylactic surgery. Why? Because BRCA1 and BRCA genes were detected. Napoleon Bonaparte, his family, all the members were suffering from the E category. It was found, and so everybody suffered from the gastric cancer. So I think the future will be no surgery, no chemotherapy, no radiation therapy. Like this, like say, nothing is impossible. Everything is possible. अब साथीहरू हामी इम्प्रोभाइज गरौँ मलाई जस्तो लाग्छ के भने बिस्तारै हामी उही कन्भेन्सनल सर्जरीमा गइराख्यो भने हामी चाहिँ ओपन सर्जरी ल्याबमा मुभ गर्यो रोबोटिक रूपमा गर्दैन भने फार बिहाइन्ड